As a physician, I wanted more and to move and to step into a more precision medicine-based approach in patients. A lot of the usual industrial partners have run away from the transplant field, and the outcomes are not perfect, so we need new drugs. And the major hurdle to develop new drugs in transplantation is that we need to follow patients for a long time before observing the true and the hard endpoint, which is return on dialysis. And we know that other endpoints developed by the Food and Drug Administration, like biopsy-proven acute rejection or just allograft function, as standalone are not sufficient. In other words, if you launch a clinical trials, you will have to wait for 10 years to have the hard endpoint, which is patient death or return to dialysis. With the iBox system, you can provide an information just 1 year after the randomization that will actually fit perfectly with what will be observed 10 years after that. The iBox score is an algorithm that will capture some usual parameters measured after transplantation, like kidney function, histology, time, presence of antibody, and will convert this information into the likelihood or the risk of allograft loss at 1, 3, 5, 7, and 10 years after the iBox evaluation. What we are looking for with the iBox is its capability to inform the inclusion and exclusion criteria, moving from expert opinion to computer assistant, whether you can interrogate your reference set, use the iBox, and inform the different scenarios that will be the best one that will fit into your hypothesis and your drug. So what is the added value of the iBox in clinical trials? 3 points. The first will be to use the iBox for the design of the trial. So it's that before the trial has started, we can contextualize and take the inclusion and exclusion criteria and draw different scenarios which will help for sample size calculation and the definition of the endpoint. The second point is to capture the true endpoint and to use the iBox as a surrogate, meaning that when the study has started, we can use the iBox assessment at many different time points after the treatment randomization and evaluate the trajectory of the patients, the differences between arms, and to have these answers at different time points after transplant and project any individual patient from the trial, not only at 1 year, but also at 7 and 10 years after randomization. So what is the robustness of the iBox system within the clinical trials? First of all, we were able to show in the multiple validations that we did in those trials that the performance of the iBox was conserved within those trials. And importantly, the trials that we have included where we tested the performance of the iBox were trials covering different clinical scenarios. Antibody-mediated rejection, T-cell mediated rejection, calcineurin inhibitor sparing strategies. So we were very pleased to show not only the conservation and the transportability of the iBox within those trials, but also the fact that we already were able to demonstrate that we had the true outcome projected by the iBox about 5 or 6 years after the termination of the study. How the iBox fill the gaps into defining surrogate endpoints, as I previously said, by being a very strong and validated prognostic score, validated in the setting of different populations, randomized control trials, and capturing the response to therapy. The iBox has many components for entering a qualification process of surrogate endpoint by the agencies.